So hello students, welcome to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Students, welcome to the channel. Now students, this is a lecture from the series NCERT line by line, where we solve every single line of NCERT. We understand the lines of NCERT to make your life easy. Okay. Now students, before we go any further, let me know in the chat if I am audible to all of you and the presentation behind me is visible to all of you. Quickly let me know in the chat. And also if audio is visible. Yes, if audio is visible, let me know in the chat right now. Good morning, good afternoon each and every one of you. Rather good morning, right? Good morning. Now students, the lecture is called as Principles of Inheritance. All of you know the chapter, right? All of you know the chapter. I'll tell you a small hack right now. This chapter is actually very easy. This chapter is actually very easy. But the concepts are very easy to understand. It's very easy to understand what is monohybrid, dihybrid, incomplete dominance, codominance, polygenetic, pleiotropy. Everything is very easy to understand. But the questions will make you think a little. Yes. So in this lecture, what we'll be doing is, we'll quickly cover the syllabus, we'll quickly cover the concepts, the line by line of NCERT. Then I'll make sure you understand which type of question you can expect from this chapter. Deal? So what we'll do right now is, we will first learn the concepts and then I will show you exactly which type of question you can expect from the monohybrid cross, which type of question you can expect from your dihybrid cross, because they will never ask you phenotypic ratio, genotypic ratio. No. What they will ask you is a certain twisted question. So if you stay till the very end, you will be able to solve the questions and also get a good understanding of the concepts. So with that being said, can we start today's amazing class? If yes, quickly green hearts in the chat, all of you. We will start today's class. Okay. Can we start, students? Yes. Can we start, students? Can we start today's class? Quickly green hearts in the chat. Okay, so what we'll be covering in today's class, students? In today's class, we will be covering Mendel's law of inheritance, which is basically three different laws: law of dominance, law of segregation, and law of independent assortment. Then we'll be learning inheritance of one gene, inheritance of two genes, monohybrid cross and dihybrid cross. Then we will also be learning, I think we will stop there. These three topics in the next class. First three topics. Clear? Let's start, students. Now, have you ever heard in your life that your eyes looks like just like your mother? Let's say Tanuja, have you ever heard that your mother, anyone says your eyes looks like your mother, or your nose is like your father, or any one of you? You might have heard, right, in your life that your physical features resembles your parents, something or other, the same similarities are there. You might have heard. Now, similarly, you always see in the nature that an elephant will give birth to an elephant. A dog will give birth to a puppy. A cat will give birth to a kitten. Okay, I was going to say kitty, but okay, kitten. Okay, so that is called as like begets like. That is, a parent to the offsprings, some kind of genes or DNA is transferred. Yes or no? So that is what we learn in the first paragraph. It is given right here. Okay, it's given right here that, one second, let me change the color. Have you ever wondered why an elephant always gives to birth to only a baby elephant? Always? Yes, always a baby elephant. Have you ever seen elephant giving rise to a giraffe? Is it possible? Elephant giving rise to a giraffe? Not possible. Okay, that will be very weird then. And not some other animal. And why a mango seed always forms a mango plant? It would be nice, from mango seed if you could get pineapple plant. Not possible, unfortunately. Okay, given that they do, there are offsprings identical to their parents. Yes, we are identical to our parents. Not exact copy, but they are identical. Because there are small changes also. Right, there are small changes also. Okay, they also show differences in some of their characteristics. Yes, we show. For example, I say my nose is like my father, but it is not always the case that my eyes will also be like my father. No, the small changes are there. Okay. Now, have you ever wondered why siblings sometimes look so similar to each other and sometimes even so different? See, me and my sister look almost alike. Everyone says. but there is always a small difference is always going to be there. Okay, small difference is always going to be there. Okay, now these are several related questions which we are dealing with scientifically. You will learn in this chapter. And also students, this entire genetics, right? This entire chapter belongs to genetics. Genetics is a branch of biology. It is a branch of biology that deals with inheritance and variation. Now students, can anyone tell me what is microbiology then? Can anyone tell me in the chat what is microbiology? Anyone in the chat? Okay, this subject deals with the inheritance as well as the variation. So genetics is a branch of biology that deals with inheritance and variation. Inheritance and variation. Now what is inheritance, students? Inheritance is nothing but the phenomena by which physical, psychological and behavioral features are transferred from the parent to the offspring. That is called as inheritance. I told you, your eyes are like your mother. Exact copy, inheritance. But can there be a small change? That change is called as variation. The small change is called as variation. Inheritance is transfer. A small change in inheritance is called as variation. It is called as what? Variation. Okay. One second, friends. An extra slide came from no reason. Okay. Now humans knew from the early 8000 to 1000 BC that one of the causes of variation was hidden in the sexual reproduction. Can anyone tell me why? Why the sexual reproduction can lead to variation? Why, students, can sexual reproduction lead to variation? Anyone? Yes, it's Diksha ma'am's birthday today. Exactly. It's Diksha ma'am's birthday, not Diksha sir. Diksha ma'am's birthday, so wish her, everyone, okay? Separately. Now students, we have a special surprise for that. Stay tuned on the channel. You will find a separate video today on the channel. We will be coming live, all four of us. So stay till the end. Today you will have a special surprise for all of you. Okay, we'll surprise Diksha ma'am also. Okay, we have a surprise for Diksha ma'am also coming up. Okay, now students, our question was sexual reproduction. Why sexual reproduction leads to variation? The answer is very simple, because in sexual reproduction, the two gametes are fusing, one from father, one from mother, and when the gametes are being formed, that is the result of meiosis. And every single time there is meiosis, there is recombination. So, and two gametes are fusing, and the zygote is the result of two different gametes. So, there is always variation. Always variation. Now, students, do you find variation? Do you find variation in the case of asexual reproduction? Can anyone tell me yes or no? True or false? Do you find variation in asexual reproduction? True or false? Quickly. They exploited the variation that were naturally present in the wild population. See, they are also abusing. They are also taking advantage. That from that time till now, we are only taking advantage of nature. Now also we are taking advantage. Cutting the plant, cutting the tree, do something. Back then also we were taking advantage of plants and animals. Okay, they exploited the variations that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that are possess the desirable characters. For example, I'll give you a quick idea. How many of you like mangoes? Raise your hands. How many of you like mangoes? Raise your hands quickly. Quickly, raise your hands. How many of you like mangoes? For example, students, there is a mango plant here. There is a mango plant here. Mango plant 1, mango plant A, and mango plant or mango tree, you can call it mango tree B. Okay, we have mango tree A, mango tree B. Okay, mango tree A and mango tree B. Now here, mango tree A, students, mango tree A has big, or large mangoes. It is producing big and or large mangoes. And this mango is very sweet mango. Sweet mango. Okay. Now the A mango tree is what? Big and large mango. And B mango is sweet, very, very sweet mango. So what do we do now? We selectively hybridize them. We selectively hybridize this plant, and what do we get? The result is what, students? The end result is going to be what? The end result is going to be a big mango which is also very Sweet. Very sweet. This is called as selective hybridization. This is called as selective hybridization. Okay, which we have been doing here. See, selectively breed and select for organisms that are possess desirable characters. For example, through the artificial selection and domestication of ancestral wild cows, we have well-known Indian breed, that is Sahiwal cows in Punjab. Sahiwal cows in Punjab, they can ask you a question, you should know. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had a very little idea about the scientific basis of this phenomena. That is, they knew some variation is there, something is being transferred, but they will never tell you. Okay, students, how many of you know, they tell in the house, right, if you cut the nail, you should not cut the nails in the night time. Does anyone know why? Friends, they always tell we should never, have you heard about it? My granny used to tell me, do not cut the nails in the night time. See, they had their own reasons, but whenever I asked the granny my reason, she's like, nah, just follow it, you will be better, it's better for your life. Okay, now after so much of intense research, I found that the reason they used to tell do not cut the nails in the night time is because back in the day they used to not never have light. So they used to cut the nails in the daytime. If they cut in the night time, they used to use the candle or some kind of fire or some kind of very dim light. So very difficult to cut the nails. And back in the days, nails were cut with the help of not a proper nail cutter. They had a blade. They used to cut with a blade. So if you cut with the blade in the night time, there is a very high chance that you cut your finger also. And with that, the tradition came along that do not cut the nails in the night time. But right now we have amazing light. We have scientific nail cutters, right? So we can cut the nails aram se. So there was some scientific reason, but we did not know about it, that's all. Okay. All of you also know the reason now. Okay. Very simple reason, but it makes sense back then. It made sense back then. Okay. So all of you know what is inheritance? Inheritance is nothing but transfer of characters. Transfer of characters from parent to the offspring is called as inheritance. And when the inheritance is happening, there are small changes. There are small changes during inheritance, and the changes is called as what? Variations. The changes are called as what? Variations are called as the changes. Very simple chapter. Okay. Now students, now we will understand about your one uncle, famous uncle, but a little bit of sad life he had, but other than that, very amazing. Okay. The uncle name is Gregor John Mendel. Let's learn about him. Okay. It was during the mid 19th century that headway was made in the understanding of inheritance. So understanding of inheritance took place in the mid 19th century. Okay. Gregor Mendel conducted hybridization experiments on garden pea plants for 7 long years. You think your life is difficult, huh? You think your life is difficult that you have to study for NEET examination for 1 year? Imagine the Gregor uncle. For 7 long years, he did the hybridization experiment. Same experiment over and over for 7 long years. That is between 1856 to 1863. And proposed the loss of inheritance in the living organisms. During the Mendel's investigations into the inheritance pattern, it was for the first time that the statistical analysis and mathematical logic were applied. Very important line. Now you might ask why? Because students, remember, Mendel actually wanted to become he was a mathematician. He wanted to become a maths teacher. But he could not succeed in that field. So what did he become? He ended up becoming a biologist. This was his second option. The first main option was to become a priest actually. He wanted to become a priest in the church. Then he wanted to become a mathematician. But then he's like, wait, mathematics is not my cup of tea. I will turn into biology. Okay. So when he failed the examination, he came back to his mom in the church, and there he started noticing. That every single plant has such amazing variations. That became very interesting to him. So students, what you need to do is look around you. If you find something interesting, try to find more about it. Okay, that is what Mendel did. Okay, that is what Mendel also did. Okay, so his apart from every other biologist, Mendel started using mathematics in his studies. That was his plus point. That's what made his research more, valuable, right? Analysis and mathematical logic were applied to problems in biology. So you might ask, sir, why always to understand why maths? Why maths? Maths is important. Okay, maths is very important. His experiments had a large sample size. Now, what is the meaning of large sample size? The meaning of large sample size is that he used a lot of plants, lots and lots of controls, lot of replicas, okay, which gave greater credibility to data that has collected, okay, that has been collected. Clear? So this is about your Mendel. This is about your Mendel. Okay. Now, where is it? Also, the confirmation of his inheritance from his experiments was successive generation of his test plants proved that the result pointed to a general rule of inheritance rather than being a unsubstantial ideas. Mendel investigated characters in garden pea plants. So, students, what did he work on? He worked on the plant that is called as pea plant. It is also called as what? Pisum sativum. Pisum sativum, the garden pea plant. The garden pea plant. Okay. That were manifested as two opposing traits. Now, example. Now, students, understand the difference between a character and trait. Understand the difference between a character. Can anyone tell me in the chat, what do you mean by a character first of all? What do you mean understand by character first of all? What do you understand? Let me see the chat, students. Are you liking the video or not, students? Have you smashed that like button? The target is 150 likes today. Extreme we are going. Next level. 150 likes. Okay. 150 likes. What is a character, students? It is the phenotypic property. It is a phenotypic. It is a phenotypic property. Phenotypic property of an individual. Phenotypic property of an individual is called as character. It is called as character. I'll give you a quick example. Example can be height of a plant, color of a flower, color. Now students, did you realize here I am not talking about tall or short. I am not talking about which color of a flower, red, blue, pink, green, yellow. No, I am not talking. Though you don't have green flowers and all, very not possible. But yeah, very rare. But this is a character. It is a phenotypic property of an individual. Then what is a trait? What is a trait, students? What is a trait? Understand. What is a trait? Understand, students. Trait is nothing but the phenotypic property plus the variations. Okay, that is inherited character plus the variations. Now, what do you understand here? Plus variations. What do I mean by this? Now, here I told about height. Now, here I will talk about its variation, tall or dwarf. Here I spoke about color of the flower. Here I will talk about which color is it. Is it white color? Is it white? Is it red? Or is it pink? Or it is blue? Now, students, do you understand? So, in the case of character, it is just a phenotypic expression of an individual. But when we talk about trait is a inherited character plus the different types of variations which come along. That is a trait. Okay. I hope this is very clear to all of you. Okay. I hope this is clear. Very simple, yet many students make mistake here. Okay. So what is a character and what is a trait? Understood, all of you? Character and trait. So next point. So here we spoke about that were manifested as two opposite traits. See, two opposite traits. There are two traits here. What are the traits here? Tall or dwarf. See, tall or dwarf, they are traits. They are not character. Okay. Green or yellow, This allowed him to set up the basic framework of the rules governing the inheritance, which was expanded on by later on scientists to account for the all diverse natural observations and the complexity inherent in them. So he took a pea plant which showed a clear difference in the traits. In a pea plant, you are able to see a plant is very tall or dwarf. You can easily clearly see the seed color and seed shape. You can easily see the flower color. Which flower color is it? Is it violet or is it white? That is an advantage of pea plant. Okay. Now, Mendel conducted such artificial pollinations or cross pollination experiments using several true breeding lines, pea lines. Students, what is this true breeding lines now? What is this true breeding line? Students, all of you smash the like button first of all. Smash the like button. You're not smashing. Okay. Now, what is this true breeding line, students? What is this true breeding line? Okay. What is true? Where do I write here? I'll write here down. Students, because there is no space there. Okay. There is no space there. What is this true breeding line? Okay. True breeding lines. The true breeding lines are nothing but plants which has undergone repeated self pollination. Repeated self pollination can lead to true breeding lines. Okay, true breeding lines. Clear? And they're mostly homozygous in nature. They're what? Homozygous in nature. For example, I'll give a quick example here. Right, students, quick example. They can be capital T, or they can be capital T, or they can be small t. See, homozygous. They are called as homozygous. True breeding lines are homozygous in nature. Okay? Homozygous in nature. Just understand this, you're good to go. Okay? Now, students, a true breeding line is one that has given here only, has undergone repeated continuous self-pollination. It's given here only. Shows the stable trait inherited and expression of several generations. Mendel selected 14 true breeding pea plant varieties. How many, students? 14 true breeding lines. 14 true breeding lines he selected. Okay, very important line. As a pair which were similar except for one character with contrasting trait. So this true breeding lines, right? Everything was separate. I'll tell you what is the meaning of separate. Wait for 2 minutes. I will show you the diagram here. Okay. Some of the contrasting traits selected were smooth or wrinkled seed. It's given here. See contrasting traits. When I say both are different, I mean you can easily tell which is which trait and which is the other trait. You can easily tell the difference. See here. Wrinkled seed, yellow or green seed, inflated or constricted pod, green or yellow pod, tall or dwarf plant. The list goes on. Basically, this is the one here. Can you see? These are the different characters. See, characters, seed shape, seed color, flower color. It's a character. These are traits. See, variation of each other is called as your trait. This is a character and trait. Understood, all of you? Understood? So how many characters did he take in total? Can anyone tell me the answer to this question now? How many characters were taken by Mendel in his experiment? Quickly go, students. How many characters did Mendel take into account? Let's count here. 1, 2, 3, 4, 5, 6 and 7. Right? 6 and 7. 7 different characters were taken by Mendel. So 1, 2, 3, 4, 5, 6 and 7. 7 different characters. Okay. And 7 different characters had a opposing trait also. Now students, in examination what will they ask you? In examination they will always ask you which of the following is dominant. They can always ask you which is dominant. Is in pod color which is dominant. There is a small trick to remember the dominant one. The trick is very simple, gravity. Gravity. Okay, gravity. Clear? Is the gravity. Now, now students, the first one where green is dominant, this is for pod color. Green is dominant for your pod color. Green is dominant for pod color. Okay, pod color. Then what is R? R is going to be here. Let me tell you. Which is R here, students? Can you see here? Round. That is seed shape. This is round, which is for seed shape. Round is going to be for seed shape. Then we have A, that is axial. That is axial is what? Flower position. Axial is for flower position. It is for flower position. Then what do we have? We have V, that is your flower color. V is for flower color. Can you see? Flower color violet. Violet, that is for flower color. Done. Then what is I? I is inflated. That is pod shape. Inflated is pod shape. Then we have T. What is T? T is for your basic tall. What is tall, students? That is stem height. Stem length. Where is tall? Stem height. Then what is Y? Y is yellow, seed color. That is seed color. One second. That is yellow. Yellow is going to be seed color. So in examination, if they ask you, students, which is the dominant trait of the 7 characters, these are your answers. Done. Thumbs up in the chat, all of you. Thumbs up in the chat if you understood this simple concept. Okay. Now students, now. Before we go to a question, I told you Mendel did something called as hybridization experiments. Can anyone tell me what are the 3 main steps in hybridization? I told you Mendel's work was in hybridization. So hybridization mainly has 3 steps. The first step is called as selection of parents. And this parent should be what? They should be 2 breeding lines. They should be 2 breeding lines. True breeding lines. Okay. There should be two breeding lines. Two breeding lines. That's the first step. Now what is the second step, students? The second step is going to be cross pollination. It's going to be cross pollination. Cross pollination, where we get the F1 progeny. Where we get the F1. That is first filial generation. Now what is the third step? The third step is going to be selfing of F1 hybrids. Selfing of F1 hybrids. Selfing of F1 hybrids. The three main steps. The three main steps in the Mendel's work, hybridization. So if someone asks you anywhere in the world, what is Mendel's work called as? Do not tell law of dominance. That is the law which we got. His main work was hybridization. All of you will remember that. All of you will remember that. Yes or no? Yes or no? Okay. Now here is a simple question from NEET 2022. Let's see how many of you will be able to solve. Let's see how many of you will be able to solve this question. Okay. Mendel studied 7 pairs of contrasting traits. 7 pair means 14 traits. Okay, 14 traits. In a pea plant and proposed the law of inheritance. Okay. He gave the law of inheritance to us. 7 characters examined by Mendel. See, 7 characters, not 7 traits. 7 characters examined by Mendel in his experiment pea plant were said to be seed shape and color, flower color, pod shape, pod color, flower position, that is axial or terminal, and stem height, that is tall or dwarf. What is the answer? What is the answer? See, statement 1 is correct, statement 2 is also correct here. Both statement 1 and both statement 2 both are absolutely correct, on point. So they can ask you such simple questions sometimes, and wait a minute, you will have difficult questions later on, which will make your brain little okay. Now students, inheritance of one gene. So before we read the NCERT lines, can we do it ourselves? That is monohybrid cross. Can we do it, students? Tell me yes or no. Here the only thing which you need to know here is what is the meaning of F1? F1 means first filial generation or first progeny. Filial progeny. Okay. See, he conducted hybridization experiment. Hybridization experiment he conducted. Okay. See, this is the entire what is given here. What we do, what are we going to do right now? It's given already here. Okay. So let's do the monohybrid cross. Let's do the monohybrid cross. Students, always there is a simple rule to write monohybrid cross or any cross. Always you should write the parents first. Always write the parents. Always you should mention the parents first. Always mention the parents. Then we mention the gametes. Then we mention the F1 generation. Second, we mention the gametes. Then we mention the gametes. Then we mention the F1 generation. Then we have something called as selfing of F1. Okay. Tell me the gametes now, students. What are the different gametes which are used here? Are they going to be homozygous or are they going to be heterozygous? Quickly go in the chat. Very simple question. Are they going to be homozygous or heterozygous? Parents are going to be homozygous. Homozygous, one will be dominant. Now, what is a dominant, students? What is a dominant? Now, before we go dominant, I think we skipped one slide somewhere. Where is the allele part? It's not given here, I'll write here only, students. Okay. So, students, write down. All of you know what is a gene? All of you know what is a gene, students? All of you know what is a gene? Yes or no? Tell me. What is a gene? All of you should know what is a gene first of all. What is a gene? So for example, students, this is the DNA. This is a DNA, right? This is your DNA. This is your DNA. Now, if I take a small segment of this DNA, if I take a small segment of this DNA, right? A small segment of DNA is called as a gene. Okay, what is a gene, students? Gene is nothing but a small segment of DNA. Gene is a smallest segment of DNA. Smallest segment of DNA is called as a gene. Then what is an allele, students? What is an allele? Understand. What is an allele then? What is an allele then? Understand what is an allele. Now every single gene will have two copies. Every single gene will have two copies. Now students, I'll ask you one simple question. Tell me, are we diploid or haploid? Very simple question. Tell me, are we diploid organisms or are we haploid organisms? You will tell me, sir, we are diploid. We have two set of chromosomes. Just like how we have two set of chromosomes, every single gene will have an alternative form. Allele is nothing but alternative form of a gene. Alternative form of a gene. So a gene will have two different forms. You might have seen the old advertisement, no? That is a 5-star advertisement. How many of you seen? There is Ramesh and Suresh, right? Twins. So your alleles are also like twins. So one gene will have two alleles. Generally, there can be more also. That concept is called as multiple allelism, which will come to later on. Okay. So Ramesh and Suresh eat 5-star and do nothing. Okay. Similarly here also, a gene has two different alleles. Now students, you tell me, have you ever noticed some twins are exactly copy of each other and some twins look very different from each other? Have you noticed that? Yes or no? Tell me. So when the two twins are which look exactly alike, here we have a term that is called as homozygous condition. In homozygous condition, the two alleles look like each other. That is, they are very same to same. Capital T or small t. See, there is no difference. This is called as homozygous condition. Okay. Now students, have you seen some twins which do not look like each other? Look, one looks very different, other one looks even more different, right? That is called as heterozygous condition. Students, do you realize I am teaching you all the basic terminologies also? Yes or no? Tell me. So this will be capital T, small t. This is called as heterozygous condition, homozygous condition, allele, gene. So I need to make sure that my students know the basics before you go to the advanced concepts. Because in the questions, they will always give you homozygous, heterozygous. Such words will come in the question. So you need to know what is the meaning of homozygous and what is the meaning of heterozygous. If you understand, quickly green hearts in the chat if you understood the concept. Very simple. Okay. So here I am writing homozygous dominant. Now what is a dominant word? What is the meaning of dominant? Now out of the two alleles, one allele will always express regardless of other allele. That is called as a dominant allele. And I always compare dominant allele with the backbenchers. Always shouting, always making some noises coming from the back. Even if some class is going on the front, from back side some noise is coming, someone is eating the food in the background or some tabla is going in the background, backbenchers. Okay. So even though teacher is there or not there, even though someone is there in the class, right, teacher is there, he'll keep quiet. But even if the other students are in the class, they don't care. Always hangama is going on. Okay. That is called as dominant. A dominant allele will always express even in the presence of recessive allele. That is called as a dominant. Now what is the dominant allele here? The dominant allele is going to be capital T. Homozygous dominant. Now we have one more parent here. The second parent is called as what? Shut up. 1 second, students. Too much space it is taking. 1 second. homozygous dominant. I'll write down only. Okay. Other one is homozygous recessive. Other one is homozygous recessive. Now homozygous recessive is like first bencher. They are like first bencher. Okay. They will always aram se keep quiet. When will they talk? They will only talk when the backbencher is absent in the class. So recessive allele is an allele which will only express itself. Recessive allele is an allele which will only express when the dominant allele is absent or not expressing itself. So here we have what? Here we have capital T, capital T. Here we have small t, small t. Homozygous condition. Okay. Now we make the gametes. Students. Gametes time. What are the gametes? Here we get capital T. Here we get small t. Gametes. Then we do the cross. What do we get? Capital T, small t. Now friends, tell me, in this, which is dominant? Capital T is dominant or small t is dominant? Capital T is dominant. So capital T phenotype will express itself. Yes. Now, before that is, this homozygous dominant, this is going to be tall. This is going to be tall. And this is going to be dwarf. Okay. Now here tell me, what will be the phenotype of this one? Is it going to be tall or is it going to be dwarf? Both the alleles are present together in a heterozygous condition. The dominant allele will always express. So this phenotype is going to remain as tall. Then we have something called as selfing of F1. That is capital T, small t into, this is your F1, right? This is your F1. So capital T small t into capital T small t. Make the gametes. What are the gametes here? The gametes are going to be capital T, small t, capital T and small t. Done. These are called as the gametes. Now what is the next step, students? What is the next step? The next step is going to be selfing of F1. We will do it here. Now for this we need to make a box. The box is called as Punnett square, named after a scientist. Punnett square. So in Punnett square we'll take the gametes, capital T, small t, capital T, small t. What do we get here, students? What do we get here? Capital T, capital T, capital T, small t, capital T, small t and capital T, small t. Okay. Now here there is one thing which you need to write down. That is called as, what is a phenotypic expression and what is genotypic expression? Understand. What is phenotypic expression and what is genotypic expression? What is going to be the genotypic expression? Phenotypic expression is always how the plant looks from the outside. Phenotype, how the muti is looking. Okay. Now tell me the muti of this. This is going to be tall. This is going to be tall. Yes or no? Tall. Capital T, small t. T is more dominant. So tall. This also will be tall. And this will be dwarf. How many tall do we have, students? How many tall do we have? 3 tall is to 1 dwarf. 3:1 ratio. Now tell me genotype. How many homozygous dominant do you see here? How many homozygous dominant do you see in that square? I only see 1. Homozygous dominant, 1. How many heterozygous dominant do you see? Heterozygous dominant. These two are heterozygous and dominant. So 2 is to homozygous. One second. Small error. How many homozygous recessive do you see? Only 1. Homozygous recessive, only 1. one homozygous recessive. Okay. Understood? Now in examination, do you think they'll give you such questions? In examination, do you think they'll give you monohybrid cross? They'll ask you to find phenotype, genotype? No. Board exam they will, but not here. Okay. So this is your entire monohybrid cross. It's given here also. Done. Okay. Now so gene and allele is given here. We discussed already. Gene and allele is done. Okay. This is capital T, homozygous dominant, heterozygous, homozygous recessive. We discussed already what is the dominant, what is recessive. We discussed what is phenotype. What is phenotype? Phenotype is how the plant looks from the outside. Genotype, how the plant looks from the inside. Yes or no? Monohybrid cross we did just now. Okay. Heterozygous condition we get in F1. This is very important here. Understand, students. Understand. One scientist name is coming up. Understand. That is Lord Punnett. Okay. So diagram is called as Punnett square. Okay. It is developed by a British genetist, Regal D. C. Punnett. Very important. Okay. This is your entire monohybrid cross. Here is a question on the board. Let's see how many of you will be able to solve this question now. See, remember students, in examination they will always ask you question based on the some form of trickery. They'll never give the question directly. Not possible at all. Okay. So let's see how many of you are able to solve this question. Go in the chat. What is the question? A cross between tall plants were made resulting in an offspring of tall and dwarf in 3:1 ratio. Okay. Cross between tall plants were made resulting in an offspring of tall and dwarf plant with 3:1 ratio. The genotype of both parent. What is the genotype of both parents? Now students, there is a very high chance that as soon as you see 3:1 ratio, you will always run and go and click one option or circle one option. That is very high chance is that you will go for option number, oh, that is not really given. Okay. Very high chance that you will go for option number 2 or 1. Okay. Now students, read the question very carefully. First of all, read the question very carefully. The question is asking you a cross between tall plants was made. Tall plants, that is a small clue. So if it is telling tall plants, can I directly remove these two options? Because here you'll get a dwarf, here also you'll get a dwarf. So can I remove these two options? Yes, I can remove these two options. Okay, these two options are gone. Second and fourth is gone. Okay, second and fourth is completely gone. Now they're telling you when you take the two parents, you're supposed to get 3:1 ratio. Okay. Now here onwards, it is a typical selfing of F1. Yes or no? Tell me. Let's take option number 3 for example. Capital T, small t, crossing with capital T, small t. What are the gametes do we get here? Capital T, small t, capital T and small t. Do the Punnett square again. Do the Punnett square. Capital T, small t, capital T, small t, capital T, capital T, capital T, small t, capital T, small t and small t, small t. 3:1 ratio. 3 are tall, 1 is dwarf. Done. Done. So such questions will come. It is very easy to do, but there is a very high chance that you might go wrong here and there. It's because of silly mistake. Okay. Okay. Let's see how many of you can do this question now. One more question in front of you. The question says, heterozygous pea plant with violet flowers was crossed with a homozygous pea plant with white flower. Okay. Violet is dominant over white. Which of the following represents the expected combination among the 40 progeny? See, students, the question is based on monohybrid cross only. Monohybrid cross. But you might think that's such a difficult question. It is not. Let's solve this question. The question is saying what is given for us. What is given? The given is heterozygous pea plant with violet flowers. So violet is heterozygous. Capital V, small v. Okay. Heterozygous violet plants was crossed with homozygous pea plant with white flowers. Homozygous white. Right? Done. Done. Any doubt till now? This is your white. Homozygous white. and this is your heterozygous violet. Heterozygous violet. Okay, heterozygous violet done. Now you're supposed to do the gametes. What do you get the gametes here? Capital V, small v. Here we get small v. Yes or no? Yes or no? Now you do the Punnett square again. Do the Punnett square. What do we have here? Capital V, small v and small v. What do we get here? Capital V, small v and small v, small v. Now you tell me, students, what is the phenotype of this one? Phenotype of this one? The phenotype of this one is going to be what? The phenotype of the first one is going to be violet. Violet. The phenotype of this one is going to be white. White. So can I tell there is 50% is going to be violet, 50% is going to be white here. 50%, 50%, 1:1 ratio. Yes or no? 50% chance of this, 50% chance of this. Now, look at the options. It also says that the expected combination among 40 progenies. So there are totally 40 progenies are there. So out of 40 progenies, can I tell, out of 40 progenies, 20 will be what? 20 will be violet and 20 will be what? White. White. So what is the answer? Answer is obviously what? See, 30 to 10, no. 20 to 20, yes. 20 produced are violet, 20 produced are white. So students, do you realize such questions can come in examination? Yes, it is from monohybrid cross only, not from dihybrid. It is from monohybrid cross. So in the examination, they will never ask you something called as, tell me right now, what is the, expected of phenotypic ratio, genotypic ratio? No. Such questions you can expect. And this question also is a 2023 NEET previous year question. Let me know in the chat right now if you have any doubt. I'm looking at the chat right now. I'm looking at the chat. Students, any doubt till now? Any doubt till now? Students, quickly smash that like button first of all. Okay. Understood? Any doubt? Beautiful. I don't think I don't see any doubt only in the chat today. All of you are on point. Okay, thunder. Yes. Yeah. Thunder attack. Pikachu or what? Okay. Okay, see, this question is easy question. I'll give you this. I call such question as laddu questions. Who? Very easy question, students. A production of gametes by parents, the formation of zygote, the F1, F2 can be understood using the what we use this, right? What is that called as? That is called as Punnett square. Punnett square. Okay. I believe all of you are able to understand till now, and there is no difficulty in understanding. Okay. Thunder attack. Okay. This is again now students, beautiful concept again. Ready for second concept now? Ready for next concept? Ready all of you? Now I will teach you something called as test cross. Test cross. Ready all of you? Yes. Test cross. Very important to understand. Very important to understand. What is this test cross? What is this test cross? First you need to understand what is a back. There are something called as back cross. Back cross. A back cross can be of two types. A back cross can be of two types. Okay. A back cross can be of two different types. A back cross can be of two types. Okay. The first type of back cross is called as your, where we take F1 offspring and cross it with dominant parents. Dominant parents. Okay. Dominant parents. The second cross, when you take F1 parents and you cross it with recessive parents. Recessive parents. Okay. Now students, when we do the F1 plus dominant parents, this is called as outcross. This is called as outcross. This is called as what? Test cross. This is called as test cross. So what are we doing in test cross, students? In test cross, we are taking the F1 progeny. We are taking the F1 progeny and we are crossing with, we are taking the F1 progeny and we are crossing with the recessive parents. Crossing with the recessive parents. Clear or not? Any doubt here? Any doubt here? Any doubt here? So we are taking the F1 progeny and crossing with the recessive parents. That is called as a test cross. That is called as test cross. Okay. That is called as a test cross. Should I write for you? One second, students. Let me see the chat right now. There, I'm not able to read the chat only. It's become very blurry little. All of you students, quickly smash that like button first of all. And have you liked the session? Yes, you have liked the session. Okay, beautiful. Okay. Now, students, what I want to say is, what is a test cross? Okay. It is a cross between. It is a cross between dominant, very important line, dominant phenotype of F1. It is a cross between dominant phenotype of F1 with double recessive parents. Now, when I say double recessive parents, it means homozygous. Homozygous condition. Homozygous condition. Is this point clear to all of you? Test cross. Test cross. Now the question is, why do we do the test cross? Next question is why? Why do we do the test cross? Can anyone tell me in the chat, why are we even doing the test cross? Is there a need? What is the need to do the test cross? Now tell me, students, whenever I ask you a question, the agenda is to find something, to find some answer. That's why you write the test also, to test your knowledge. Yes. Now the reason we are doing the test cross is to find the genotype of F1. Why? To find the genotype of F1. To find the genotype of F1. Now what do I mean by genotype of F1? Now students, you tell me, if we have dominant phenotype in F1, we have dominant phenotype in the F1, what are the two possibilities with respect to genotype? I'll repeat the question again. Now you're little advanced, I'm telling you. The F1 progeny shows a dominant phenotype. What are the possible genotypes? For example, flower color. If I take flower color, students, I'll write here, okay? I'll write here. If I take flower color and if I tell violet is dominant, if I tell violet is dominant, if I take flower color, students, if I take flower color and if I tell violet is dominant, are the only two possibilities is capital V or capital V small v? Yes or no? Tell me. Yes or no? All of you tell me. If I'm telling the dominant phenotype is violet flower, it can be capital V or capital V small v. It can be homozygous dominant or it can be heterozygous dominant. Now you tell me, is there a possibility? Is there a possibility that it can be small v? Tell me. Can we have the dominant phenotype to be small v? Is that possible? Obviously not. If it is small v, it is going to be white flower. It is going to be white flower. Okay. So here is a thing which is given your NCERT. Hope it is visible to all of you. Okay. Now see here. What do we have here, students? Here we have the dominant phenotype. Yes or no? And why are we doing a test cross? We are doing a test cross to find the genotype, unknown genotype of F1. Unknown genotype of F1. Yes or no? Okay. Now we are taking dominant phenotype. The two possibilities here is, I don't know why they are given W, should be supposed to be V, but the two possibilities, homozygous dominant or it can be heterozygous dominant. Any of the two possibilities can take place. Yes or no? So in the test cross, we are trying to find what is the genotype. What is the genotype of F1? That is dominant phenotype. How do? We take a cross. We take a cross between dominant phenotype with homozygous recessive parents. Homozygous recessive parents. Done. Now you do a simple cross. Okay. Here we are taking heterozygous dominant. Capital W, small w. Small w, small w. Do the cross. When you do the cross, we realize that we are able to get in the product of F2. In the product of F2, what are we getting? Half of the flowers are violet and half of them are white. Unknown genotype is heterozygous. Why do I say heterozygous? The only reason we are able to get 50-50. The only reason we are able to get 50-50 is because the unknown genotype is heterozygous. Now, what if the unknown genotype is homozygous? If unknown genotype is homozygous dominant, that is, we will end up getting all the flowers are violet here. Why? Because dominant allele, dominant allele, dominant allele, dominant allele, dominant allele. So what do you understand test cross? In test, I'll write a line for all of you. Beautiful line for all of you students. Understand this line. Okay. Understand this line, all of you. In test cross, we are finding unknown genotype of F1, which is a dominant phenotype using the results obtained, which is basically the phenotype in F2. Using the result obtained from F2 cross. Using the result of F2. Did all of you understand or not? Tell me. Here we are trying to find the unknown genotype. Using the result we obtain in the F2. If the F2 has two dominant and two violet and two white, we'll automatically tell it is heterozygous dominant. If we are getting all of them are violet here, it means that result is this F2 result. We are getting the genotype of F1. That is your test cross. Done all of you. Tell me, is it clear to all of you now? It becomes very clear to all of you with the help of this statement right here. In test cross, we are finding unknown genotype of F1, dominant phenotype, using the result obtained from the F2. Using the result which you obtain in the F2. Take the result, get the test cross. Okay. Now students, see this question, all of you. See this question. Let's see how many of you are able to solve this question. I'll give you exactly 10 seconds to solve this question. 10 seconds to solve this question. 10 seconds. 10. I'll give you the question for you. It's a simple match the following. Two or more alternative form of allele is called as what? Of a gene is called as what? It is called as an allele. A is 1, so A is 3. Alternative form of gene is called as an allele. Okay. 10 seconds. Answer I want. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. Students, look at the options. Is there only a single option where it says A is 3? Is there a single option which says A is going to remain 3 as option number 2? Done. Look at this question. in a plant, black seed color, capital B and capital B small b, that is homozygous dominant and heterozygous dominant, okay, is dominant over white seed, that is small b. In order to find the genotype, we are trying to find the genotype of the F1, trying to find the genotype of the F1 dominant, okay, genotype of black seed plant. With which of the following genotype will you cross? Students, how do you find a test cross? In test cross, we take the F1 and we cross it with recessive parents. So this is your F1. F1 should be crossed with recessive parents. So which is recessive here? Small b is homozygous recessive. So answer is A, obviously. Answer is A. Students, do you want these notes? That is the bigger question. All of these notes will be available on the Telegram channel. This is my Telegram for all of you who have been asking. Okay. All the notes will be available here. And also on Vedantu NEET English Telegram channel. Both the places, notes are updated after the class in 1 to 2 days. Maximum 1 day. Okay. Maximum 1 day. Okay, students, with that monohybrid cross is over. So from the monohybrid cross, did we understand something called as law of dominance and law of segregation? So from monohybrid cross, the two laws which we obtain is law of dominance and law of segregation. Now what about law of independent assortment then? Law of independent assortment which we get from dihybrid cross. Wait for that. Okay, we have to wait for that. Okay. Now what is law of dominance and law of segregation? Very important questions you can get from here. All of you easily understand law of dominance, but some students face it difficult to understand law of segregation. I will make it easy for you. That's why they call me easy sir. Okay, I'll make it easy for you. Understand. What is law of dominance? Every point is important, students. Every single point is extremely important here. Law of dominance basically gives you 3 main points. Characters are controlled by discrete units called as factors. That is basically gene. So factors are controlled by, characters are controlled by factors. First point clear to everyone? So characters are controlled by factors. First point. Factors occur in pair. Genes occur in pair, right? 2 alleles are there. In a dissimilar pair, 2 alleles are dissimilar. Okay, that is 2 alleles are dissimilar. That is capital T, small t. In a dissimilar pair of factors, one member of a pair dominates the other. So one is dominant and one is recessive. One is dominant and one is recessive. Okay. The law of dominance is used to explain, see, very important line again. Very important line. Very important. The law of dominance is used to explain the expression of only one of the parent character. Only one of the parent character is expressing in a monohybrid cross in the F1. So did we get in F1 progeny? In F1, did we obtain only one? Yes or no? In F1 progeny, did we obtain only one phenotype? Only one phenotype, one parental phenotype, that is tallness. Yes or no? Yes. Only one we obtained, right? Only one we obtained. Okay, and expression of both in F2. So in F2 we had both. In F2 we had tall and dwarf, both are present in F2. Okay, it also explains the proportion of 3:1 obtained at F2. That is your law of dominance. Don't just say that law of dominance is only this point. No. Law of dominance doesn't just say the third point. Law of dominance also mentions the other 2 points also. So because in examination, this year examination, there was a question. Can you see here? This was the question, see, all of you. The question is very simple actually. It says, which of the following can be explained as the basis of law of dominance. Okay, out of one pair of factors, one is dominant, other is recessive. Yes, obtained. Alleles do not show any expression and both characters appear as such as in F2. Alleles do not show any expression. Is it true or false? Obviously false. Alleles show expression, yes or no? Alleles will show expression. Gone. Factors occur in pair in normal diploid plants. Yes, they occur in pair. That also is a point. That also is a point there. Occurs in pair. Okay. The discrete unit controlling a particular character is called as a factor. Yes, this also is under your law of dominance. Expression of only one of the parent character is found in monohybrid cross. Yes, in the F1 we only have tall plants. That is capital T, small t. Only tallness is expressed. So can I tell all the points belong to law of dominance, not just the first point, So law of dominance explains all the following points. So can I tell the answer is supposed to be what? ACDE 1, ACDE. All of you understand now. All of you understand law of dominance. Law of dominance. Now what is law of segregation? What is law of segregation, students? What is law of segregation? Very simple. Law of segregation is very simple, students. It tells you that alleles do not show blending. Main point. The main point is what? The main point is that alleles do not show blending. Do not show blending. Now what do I mean by that line? What do I mean by that line? That alleles do not show blending at all. What is the actual meaning of it? Understand. For that we should go back to here. We should go back to monohybrid cross. Because this law is derived from monohybrid, right? Derived from monohybrid, yes or no? See. The meaning is that the alleles do not show blending. It tells you that, for example, here we had two different alleles. Two different alleles are there. Two different alleles are here, yes or no? One dominant allele and one recessive allele. If these two alleles went under blending, it means that you cannot separate them out. Yes or no? Blending means, so imagine you're trying to make a juice or milkshake. You're adding milk, you're adding chocolate syrup, you're adding ice cream and nicely making a juice. Once you get the juice, students, can you remove the ice cream from it later on? Can you remove the chocolate syrup? Can you remove the chocolates from it again? No, right? If you're making Oreo milkshake, can you remove the Oreo after making the milkshake? Is it possible? No. Blending means you cannot retrieve it back. Blending means it is not able to separate again back. But here, alleles do not show blending. We are not making Oreo milkshake here. Alleles do not show any form of blending at all. So, what is the meaning of that? If alleles are not showing blending, we are again able to get the phenotype of the parent. If there was blending, students, if there was blending, there would no way possible that we would obtain dwarf again. Tell me, is it possible to get the Oreo back? Obviously not. This is your Oreo. Can you get the dwarf back? No, you cannot get it back. If there was blending, since alleles do not blend, alleles are not blending, we can always get the parental feature again coming back in the F2 generation. Even though the dwarfness was absent, even though the dwarfness was completely absent in the case of F1 generation, it is coming back again in the F2 generation. It is coming back in the F2 generation. That is called as law of segregation. Did all of you understand? If you understood, green hearts in the chat, all of you. Green hearts in the chat. All of you. So alleles do not show blending. They are getting the parental feature again back. Now students, law of segregation is also called as law of purity of gametes. It is also called as law of purity of gametes. Now what is the meaning of that? What is the meaning of purity of gametes? Very simple. Very simple. When in a hybrid, students, each allele separate out during gamete formation. Alleles separate during gamete formation. Gamete formation. Are they segregating separately? Law of segregation. Now, what is the meaning of purity of gametes? The meaning of purity is that each gamete is pure for a trait. That's all. That's all, students. Understand. Each gamete here, each gamete is pure for a trait. Each gamete here, each gamete is pure for a trait. This gamete is pure for dwarfness. This gamete is pure for tallness. This gamete is pure for dwarfness. This gamete is pure for tallness. From this gamete, can you get dwarfness? Not possible. Not possible. So it's called as purity of gametes. Each gamete is pure for a trait. Each gamete is pure for a trait. For a trait. That is called as purity of gametes. Purity of gametes. Did all of you understand law of segregation? Understood law of segregation? Yes or no? Understood? See, the law is based on the fact that alleles do not show any blending. Okay. And both characters are recovered in the F2 generation. In F2 generation, we are getting tallness and dwarfness, both are coming back. Both are coming back. Okay. Though one of them is not seen in the F1, that is, even though the dwarfness is not seen in the F1, it is again coming back in F2, dwarfness, because there is no blending. Though the parental contains two alleles during gamete formation. During gamete formation, parent has two alleles, capital T, small t, two alleles. Okay. During gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. This is segregation. One of the two factors is in gamete, is in the gamete. That is the meaning of this line right here. Okay, two factors. Of course, a homozygous parent produces all gametes that are similar, while heterozygous one produces two kinds of gametes, each having one allele, with equal proportion. Beautiful lines of NCERT. You are able to understand now. Beautifully, you are understanding, all of you. Done? Yes or no? Done. That is called as law of segregation. Now, friends, you need to understand that law of segregation is universal. Law of segregation, this one right here, there is no exception. Law of segregation is universal. It is universal. The meaning of universal is that there are no exceptions. There is no exception at all to law of segregation. Every single organism has to follow. No exception. But your law of dominance, students, law of dominance has certain exceptions. There are certain exceptions of law of dominance. Now, what are the exceptions to law of dominance, students? There are two main exceptions to law of dominance. That is, the first one is called as your incomplete dominance. One is called as incomplete dominance. The other one is called as codominance. It's called as codominance. So incomplete dominance and codominance are the two exceptions of your law of dominance. Now, what exactly happens in incomplete dominance, friends? What exactly happens in the incomplete dominance? Understand, friends, understand. In the case of incomplete dominance, the dominant allele is not so dominant. It is not able to completely suppress the recessive allele. The dominant allele is not able to completely suppress the recessive allele. So in that case, even the recessive allele, I will also express. So recessive allele also starts to express little bit. Okay, that is called as your incomplete dominance. So what is happening in incomplete dominance, friends? In the case of incomplete dominance, one allele is incomplete dominant. One allele one allele is incompletely dominant over the other allele. It's called as incomplete dominance. It is called as your incomplete dominance. Now, as a result of incomplete dominance, okay, as a result of incomplete dominance, there is a change in the offspring now. Okay, F1 generation, there is a change in F1 generation. F1 generation do not resemble both the parents. Is that completely unique? It is completely unique in nature. The parent is red flower, white flower. The offspring is going to be, F1 is going to be pink flower. For that we have a cross. Students, called as incomplete dominance cross. Can we do that now, students? Can we do that now? Yes or no? First we have parents here. Always write on the parents first. Parents. Then you write the gametes. Then you write the F1. Then we have selfing of F1. Okay. What are the parents here? Parents should be always what? Homozygous? Yes or no? Homozygous dominant and homozygous recessive. We have red flower here, red flower and white flower. This is homozygous dominant. So it should be what, students? Capital R or small r? Tell me. It should be capital R or small r? Homozygous dominant, students. It is supposed to be capital R. Homozygous dominant. This is small r. Homozygous recessive. What are the gametes here? Capital R, small r. F1, what do we get, students? In F1, what do we get? Capital R, small r. Now, students, typically, what is supposed to happen? Typically, this is supposed to be red. But since this is called as incomplete dominance, the redness is not able to completely go over the whiteness. This capital R is not very dominant. Incomplete dominance. So, as a result, we will get pink flower here. Pink flower will get here. Now, what do we have? Then we have something called as selfing of F1. Then we do capital R, small r, gametes, crossing, gametes, capital R, small r, capital R and small r. Now we do the Punnett square. Students. Let's do the Punnett square. What do we have here? Capital R, gametes, no? These are gametes. Capital R, small r, capital R, small r. What do we get, students, now? This is capital R, small r, capital R, small r, and small r. Now tell me, phenotypic ratio. The phenotypic ratio is going to be what? Red. These two are pink, white, pink, and red. What is going to be a genotypic ratio? 1 is homozygous dominant, 2 are heterozygous dominant, one is homorecessive. So can I tell students, in the case of your incomplete dominance, phenotypic ratio is equal to genotypic ratio? Yes or no? Both are equal to equal? Equal. Phenotypic ratio is equal to genotypic ratio, just like your test cross. That is the beauty of incomplete dominance. But in examination, how will they ask you question? Students? In examination, they will never ask you ratio directly. They'll never ask you this. In examination, they can ask you in which organism this is found. The plants which we find here is something called as snapdragon or atrium species, or mirabilis jalapa, or 4 o'clock plants. Okay, it's given here. So they can ask you the species where it takes place. Okay, see, snapdragon or atrium species. Okay, good example of incomplete dominance. The same thing what we did here is written here only. You can read it once. See, 1:2:1 ratio. 1:2:1 ratio is obtained. Okay. See, 1:2:1, 1:2:1 phenotypic genotypic ratio. Let's solve the question now. Let's see how many of you are able to solve this question now. Time starts now. I'll go drink some water and come. All of you. Solve this question. It's a NEET 2024 question. Okay. Friends, let's solve the question now. Can we solve the question now? What does the question say? It's saying a pink flower snapdragon was crossed with red flower. Pink flower is crossed with red flower snapdragon. What type of phenotype is expected in the progeny? So pink flower, okay. Pink flower is crossed. Capital R, small r, crossed with red flower. Capital R. Gametes. Punnett square. Phenotype. What is the answer? Red flower as well as the pink flower. Red flower as well as the pink flower. A is the answer. Next question. In Antrium species snapdragon, a red flower was crossed with a white flower, and in F1 generation, pink flower was obtained. Okay. When pink flower was self, the F2 generation showed white, red, pink flower. Choose the incorrect statement from the following. Let's see who can solve this question now. Okay. The ratio of F2 is 1:2:1. Okay. The pink color in the F1 is due to the incomplete dominance. Yes. The experiment does not follow the principles of dominance. Yes. The law of segregation does not apply to the experiment. No. Why? Because law of segregation is said to be what? It's said to be universal. So this is the wrong statement. Wrong statement right there. Wrong statement. Next question. Andalusian fowls have two pure forms, black and white. Okay. If black form capital B and white form WW are crossed, F1 appear to be blue color. F1 appear to be blue color, capital B small w. Due to incomplete dominance, which of the following would you be an outcome of cross between black form and blue form? Black form and blue form. Students, very easy question again. Very easy question. If you do not think and directly like, oh, incomplete dominance, so answer should be 1, you will go wrong. You will go wrong. Okay. So read the question carefully. The question is saying it is a cross between black form and blue form. Black form is going to be WWBB and blue form is going to be what? Blue form is going to be capital B, capital W. Gametes. Done. Cross. This is black form. This is blue form. So what is the answer, students? What is the answer now? What is the answer? 1 black form, 1 blue form, 1:1 ratio. 1:1 ratio. Students, are you getting an idea? Are you able to understand which type of question you can expect in examination now? They will never ask you simple questions as phenotypic ratio, genotypic ratio. No. Such questions will come. And if you do not understand or you're not aware of such questions, there's a very high chance that you'll click on 1 and it'll come out. Okay, you'll click on 1. So answer is obviously 4, 1:1 ratio. Done? Done? Got it, all of you? No. Now next there is a concept called as concept of dominance. Okay, why the dominance even happens? Why is there called as something called dominance? Okay, very easy to understand, but they usually don't ask you this examination. Okay, they usually don't ask you this concept of dominance, but let me explain to you. Because line by line of NCERT, no? Friends, so students, if you're able to enjoy the line by line still, see, I know when the line by line started, all of you had super josh, super josh was there. We'll do line by line NCERT. But as the series progressed, the energy is decreased. But here we are still doing it. It shows our commitment to the series and our commitment to the YouTube as a platform, right? So we could have just said, let's cancel this and we'll go home, but we didn't do that because we know our students depend on us for the line by line of NCERT. That is why we are making sure that every single chapter is covered here, right? So students, if the same enthusiasm is not followed, it becomes a little demotivating also, right, as a teacher. So I want everyone to always show the support from your side, like the video, let us show your love in the comments, show your support in the comments. It also makes us happy and motivate us to teach all of you in a very enthusiastic manner, which will benefit all of you. Okay, right? So concept of dominance is very simple again. Concept of dominance is very simple. For example, here we have a gene, students. Here we have a gene. A gene can give rise to a protein. The protein will express. Usually we have a gene as 2 alleles, right? For example, you have a capital A and small a. Now students, the reason why this is different. The reason why this is different is, apparently, according to concept of dominance, the reason we have a recessive allele, this is because of certain modification. Now, what modification? The modification which they are talking here is basically some form of mutation. Some form of mutation would have taken place. And that is why we have the recessive allele. Now in NCERT they are giving you example also. They are giving you an example here. Okay. Where is the example? Let us take a gene that contains information for a particular enzyme. One gene is there, it has an enzyme, it has the information for enzyme. The two alleles forms. Let us assume that as in more common, the normal allele produces normal enzyme that is needed for the transformation of A to S. Theoretically, see, this is normal. This is normal. This is normal. This is normal. For example, this is producing normal enzyme. Normal enzyme. This is producing a modified enzyme. Modified enzyme. Okay. Modified enzyme. Now, theoretically, the modified allele would responsible for the production of the modified allele. The modified allele can give you a normal or less efficient enzyme. It can give a non-functional enzyme, or it can give no enzyme at all. So when these two cases happens, it mainly results in a recessive allele. Recessive allele, which is also modified. This one may be modified, but it is acting like a dominant allele. Dominant allele. Okay, so it's given here. In first case, modified allele is equivalent to unmodified allele. That is, it is producing a normal enzyme only, so it is equivalent to unmodified allele. Both are same to same. Okay, it will produce the same phenotype trait, result in the transformation of substrate S. Such equivalent allele pairing are very common, but if the alleles are non-functional, the second option, non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the modified allele. The unmodified functional allele, the first one, which represents the original phenotype, is dominant. I told you already. And the unmodified allele, generally recessive allele. Hence, in the example above, recessive trait is seen due to non-functional enzyme or no enzyme at all. So the whole point of this concept of dominance, is that the recessive allele came into existence. The recessive allele came into existence because of a modification, mutation. That's all. Okay. Now next concept is called as codominance. Friends, I will teach you for next 30 to 40 minutes maximum. 30 minutes you can give me of your time. 30 minutes, today's class is over. I do not want to keep the class for 3 hours, 4 hours and all. That we'll do later on. Okay. So 30 more minutes or 40 more minutes maximum, today's session is over. So I show some more energy. All of you run and go drink some water and come. Run and go drink some water and come back to the class quickly. No break. Okay. The next concept is called as codominance. So very amazing concept. I have always fascinated by codominance. Okay. So what exactly is happening in codominance, friends? Okay. What exactly is happening in the code? Hello Mikasa, welcome to the class after a long time. Okay. See, in codominance, what is happening is, in the case of codominance, both alleles are dominant. There is no concept of backbencher here. There is no concept of front bencher here. The whole class is backbencher. Here. Both alleles are dominant here. Both the alleles are completely dominant. As a result of that, the F1 generation will resemble both the parents. Will resemble both the parents. It look like father also, it look like mother also. Okay. I drank some water and ran. Okay, don't worry. Now, till now we have discussed crossing over where F1 resembles either of the parents' dominance or in between incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. Okay. Example. Now, what is an example, students? Students, the laugh comes automatically, you know. There is students in the class. Have you noticed? There is that one student who will laugh at some stupid joke also. who will laugh at every possible situation. I am like that. I am that student. I am that friend who will get you in trouble actually. Okay, who will laugh at random things. Okay. Now students, understand here. Codominance, understand. Codominance, give an example is given. That is your ABO blood grouping. That is your ABO blood grouping. Okay. That is blood grouping in the case of humans. Okay. ABO blood grouping are controlled by a gene that is called as I gene. Okay. So blood grouping is controlled by students. This is your codominancy. Here one trait is there. Mother, father, child will have both. Child will have both of them. Both of them. Okay, that is your codominance. So what exactly happening in codominance, students? In the codominance, okay, you need to understand there is a concept called as multiple allelism. How many of you heard about it? Multiple allelism. Multiple allelism. Multiple allelism. I'm not able to see, so much of reflection is there. I'm not able to see what I'm writing only. Multiple allelism. So much of reflection. Okay. Now what is this multiple allelism, students? You're not liking the session, students? Okay. Sukanya is like directly when message is coming from students. Again we are telling, Tejas series is a very special bio series. Very special bio series. And we can stop it anytime if there is no, if we think that students are taking for granted and they are not actually caring about the series. Okay. There was no rush from anyone to start a bio series. In biology we have covered the entire syllabus twice. So Tejas series was very special because it was Gopika ma'am's personal decision. Both of us took a personal decision. Then we spoke to Shreyas sir. Shreyas sir approved, and we got a special bio series for all of you. So students, energy should be very high there. Do not disappoint us there. Okay. Now students, multiple allelism. What is the meaning of multiple allelism? Okay. All of you know till now, allele has two forms, Ramesh and Suresh. We always have Ramesh and Suresh, two alternative forms. But in the case of multiple allelism, what happens is more than two alternative forms will be there. More than two alternative forms will be there in the case of multiple allelism. Many alleles. Like you might have noticed, in the case of humans, there are maximum 4 blood groupings, A, B, AB and O. But in the case of cow, there are more than 800 blood groups. Why? Because of multiple allelism. More than 800 in the case of cow. Okay. So that is called as multiple allelism. Okay. There are 3 different alleles here. 3 different alleles. 3 alleles are there. That is IA, IB and small I. These two are what? These two are dominant alleles. And this is recessive allele. Papa. One recessive allele is there. Okay. Now students, even though there are 3 alleles, students, even though there are 3 alleles, only 2 will be present. 2 will be present in the human body. Just because 3 is present, all 3 cannot be there. Just because 3 are present, all 3 cannot be present in your body. Not possible. Why? Because we are diploid organisms. We are 2N. Right? Meaning we have 2 set of chromosomes. One chromosome is coming from father, one chromosome is coming from mother. Yes. Similarly, genes also occur in pair. Alleles also should be in pair. One allele should come from mother, one allele should come from father. That is always the case. You cannot have 3 alleles from somewhere, one more allele is coming. Not possible. So just how we are diploid, 2 set of chromosomes, alleles are also present in only 2 in our body. One from mother, one from father. Okay. One from father, one from father. Okay. Now students, I want a volunteer from the chat now. I need a volunteer. Whose blood group I want? Whose blood I want? Or blood group. Who is the volunteer? Who is the volunteer? Ranveer. Who is the volunteer, students? Who is the volunteer? Students, Gopika ma'am is in the chat. Students, I'm telling you again. Tejas series, something here and there if you do, gone it is. Okay, let's take Mikasa only. First person to volunteer. So, Mikasa, I want your blood group. What is your blood group? Tell me quickly. What is your blood group? What is your blood group? Blood group. Anyone? Blood group. Blood. We need blood here. We need blood. O positive. O is not very fancy. We'll choose something. Do give me. O is not very fancy. O is very simple. Just like we need some fancy blood group. Okay. We need one fancy blood group. Tell me. Someone has B. Someone has AB. Someone has A positive. Anyone? Okay. Let's take. Okay. Blood group only changed. Okay. Let's take Ranveer. Ranveer is saying A positive. Okay. Ranveer is saying A positive. So Ranveer is saying A positive. His blood is the alleles to be present is going to be IA or I, or it can be IA or IA. These are the two possibilities of the A blood grouping. A blood grouping. Both are dominant. A blood grouping. One is dominant, one is recessive. A blood grouping. Anyone with AB? Okay, AB is a Sony is there. AB. In the case of AB blood grouping, they'll have IA and IB, the only possibility. B blood grouping, we can have IB, or we can have IB or I. In the case of O blood grouping, universal donor. Okay, they will have IA, I and I. Clear? Clear? This is the blood grouping which is given in your syllabus. Now students, why do we think IA is so what is the difference here actually? Why do we call it IA as different, IB as different? What is the main reason for the difference? The main reason for difference is the sugar actually. If you look at your hemoglobin, right? If you look at the hemoglobin on the hemoglobin, on the antigen, you can see some form of sugar. Okay, sugar. Okay. Along with H antigen, along with HC, this is H antigen. H antigen plus there will be some sugar will be there. In A blood grouping, in the case of A blood grouping, it is N-acetyl galactosamine. It is N-acetyl galactosamine here. N-acetyl galactosamine. Now, what about in the case of your B blood grouping? In the case of B blood grouping, students, in the case of B blood grouping, there is galactose here. Can you see? There is galactose here. Galactose. Okay. There is something called as galactose in the case of B blood grouping. Now, because of this sugar, there is always a change. Okay. See, it is given here, which I told you. The line which I told you is given here. Because humans are diploid organisms, each person possesses only any two of the three alleles which is given. Okay. Done. This is a table already done. I will be I. Only this is also done. Multiple allelism also done. More than two alleles called as multiple allelism. Solve this question, students. Solve this question. So as you will see, students, theory is very easy. Theory is very easy to understand. The main thing which you need to focus on the questions. The questions on the board, you go. Where am I from? I'm from I'm from Bangalore. Yes, I'm from Vedantu. Wow. Wow. Sir is from Vedantu. Tell the answer quickly, all of you. See, as per ABO blood grouping system, the blood group of father is B positive. Father is B positive. Mother is A positive. Okay. And the child is O positive. Child is O positive. Okay. Their respective genotypes can be. What is the respective genotypes? What is the respective genotypes? Students, if the child is O positive, he or she has to be II. Yes or no? INI. So can I immediately remove this option? Remove this option? Remove this option. It's either of these two can be answer. Either of these two can be the answer. Okay? Now, father is B positive. Okay, B positive. Mother is A positive and child is O positive. But in the second case, can you see here? It is IB, IB and IA, IA. Both are dominant. There is no possibility there will be II. The child will be here what? The child will be here IA or IB. That is a possibility there. That is a possibility. You cannot have II. The only answer is option number 1. The only option is answer number 1. Okay. So only A. Only A. Laddu question from NEET 2024. See students, if you think there are difficult questions, yes, there are some questions are difficult, but there are also very easy questions are also there. Yes. Dinesh, calm down, calm down. It's fine. I can see the answer. Once also if you type, I'll see the answer. 10 times if you type also, I'll see the answer. Okay. ABO blood grouping is an example of multiple allelism. Multiple allelism. Okay. Multiple allelism. Calm down. Next question for all of you. Next question for all of you. Next question for all of you. Identify the wrong statement with reference to I gene that controls the ABO blood grouping. First option only looks suspicious. First option only looks suspicious, students. Sus. Do they have same type of sugar? Do they have same type of sugar? No. Two different type of sugar? No. So, wrong statement. Students. Now, this paragraph is usually ignored. This paragraph is usually ignored, but we will not ignore. Why? Because we are doing NCERT line by line. Are we allowed to ignore? No, we are not allowed to ignore. You cannot ignore this at all. This paragraph is very special because very difficult to understand. Okay, very difficult to understand. Okay. Now, students, just read this last paragraph. I'll give you 2 minutes. I'll go drink some water and come. All of you read this paragraph and tell me if you can understand. Read this paragraph and tell me if you can understand this paragraph. Tell me. Just read this paragraph and tell me if you're able to understand anything out of this paragraph. You gotta tell me. I'll just go drink some water and come. Read this paragraph. Students, do all of you understand? Tell me. Understood? Very good. We'll go to the next concept now. Understood? Can we go to the next concept? Obviously not. Okay, obviously not. Students, understand there is something called as everyone is very special. Yes or no? Now you tell me, just because all of you, I'll give you a quick example, understand? Okay. All of you, I'll give you a quick example, understand? Just because you are good in, cricket, does it mean that you will also good in football, chess, badminton, table tennis, swimming, everything? Tell me. Everyone has a stronghold. Just because you have good memory, does it mean that you will be good in bio, physics, chemistry, maths, computer science, astrology, astrophysics? You will be good in everything? Tell me. Yes or no? Tell me yes or no. Just because if you are good in one thing, does it mean that you will be good in everything? Not possible, right? Not possible, right? Same thing applies to the concept of dominance. Just because one allele is dominant for one trait, it doesn't mean that allele will be dominant for all the traits. It'll be for all the traits. No, right? Not possible, right? Not possible. So understand the point here. Understand what I'm trying to tell you here is, occasionally, they're giving you an example here. They've given you an example. Occasionally, single gene product may produce more than one effect. Obviously, a gene can have more effect, right? A gene can have more effects. It can have more effects. Yes. For example, starch synthesis in pea seeds is controlled by one gene. Okay, it's controlled by one gene. Starch synthesis is controlled by one gene. Okay, it has two alleles, capital B, small b. Starch is synthesized efficiently by capital B. Nicely, starch synthesis is happening. Okay, and homozygous, and therefore large starch grains are produced. Okay. In contrast, small b homozygous have lesser efficiency in starch synthesis and produces smaller grains. Okay. After maturation of the seed, capital B seeds are round also. See, they are round also. Very important line. Capital B is round and also producing large seeds. Okay. I'll write here on top. Capital B is producing what? First of all, large starch and also round seed. So round seed. Small b is producing what? Less starch. Okay, less starch and wrinkled seed. Wrinkled seed. Okay. The heterozygous produce round and so B seems to be dominant. Okay, B seems to be dominant, but the starch grain, see, now what is the heterozygous condition? Now you cross it. When you cross it, we get what? Capital B, small b. Technically, what are we supposed to get? Tell me, students. Technically, your capital B, small b is supposed to be what? Technically speaking, it is supposed to be large starch and round seed. Yes or no? Why? Because this is dominant. In F1 generation, it has to be dominant phenotype. Okay. But what is happening here? Instead of, okay, but the starch grain produced are intermediate in size in capital B, small b. Okay, let's see what is happening here. Instead of getting large starch and round seed, what are we getting? What is the reality? The reality is that we are getting intermediate seed and round seed. and round seed. See, this gene is dominant for round. It is dominant for round. But does it also mean that it is also dominant for the stars in this also? No. Here it is not dominant. So as I told you, just because you're good in cricket, it doesn't mean that you'll be good in every sport. Chess, swimming, frisbee throw, swimming, horseback riding is not possible. Just because one gene is dominant for one trait, it doesn't mean that the same gene will also be dominant for other trait also. Okay. So the starch grain size is considered as a phenotype. Then for this angle, the allele shows incomplete dominance. It is showing incomplete dominance here. Understood? So it doesn't mean that a dominant allele will be dominant for everything. Not possible. It will be dominant for few things and incomplete dominant for others. Okay? Incomplete dominant for others. Understood, all of you? Understood? Yes, I know it is birthday. Did all of you understood? This is what we are getting. This is what we are actually getting. Okay? See here. Therefore, dominance is not an autonomous feature for a gene or the product that it has information. It is not automatically dominant for everything. It is not automatically dominant for everything. Right? See here, simple words. Just because one version of a gene allele is dominant for one trait doesn't mean it will be always dominant for everything else. If a gene affects more than one feature, dominance can behave differently depending upon which feature you're observing. If you're observing shape of the seed, yes, it is dominant. But if you're observing the starch synthesis, it is not dominant. It is not dominant here. For example, the same gene might be dominant for seed shape, but not completely dominant for starch grain in size. Not dominant there. Okay. This means that concept of dominance depends on how the gene product works and which trait we choose to focus on, what we are focusing on. Just because, imagine if I am good at cricket and I don't play cricket at all, will I automatically become good in football? No, not possible. Not possible. Okay. So very important lines, understand this concept. Very important. Now students, with that, today's final concept. Okay. Today is the final concept for today. After this you can run home. If you are home, go sleep off if you want. Now we will be doing something called as dihybrid cross. Show some energy in the chat for the last concept for today. Next class we will be doing something called as chromosomal theory in your inheritance. We will be starting from the next class. Done? Done students? Last concept for today is dihybrid cross. Very simple. I have some numericals also. We will solve that also. Okay. Let's understand dihybrid cross. The first point which you need to understand for dihybrid cross is that in the case of dihybrid cross, there are not one character but two characters. There are two different characters here. Two different characters. Okay. Here, two characters involved. Two characters are involved here. Two characters involved. Let's start parents first. Always same pattern. Parents, gametes, F1, selfing of F1. What are the parents here, students? Homozygous. Parents is going to be capital R, capital R, capital Y, capital Y. That is seed shape and seed color. Okay. Seed shape, which is round, is dominant, and seed color yellow is dominant. Seed color, seed shape and seed color. Seed shape and seed color. Okay. Round and yellow. We are crossing with small r, small y, small y. That is wrinkled and green. Wrinkled and green. And green. Okay. What are the gametes do we get here, students? What are the gametes? Gametes time. Capital R, capital Y, capital R, capital Y, small r, small y. Cross F1, capital R, capital Y, small y. What is the phenotype here? The dominant phenotype. That is round and yellow. Students, all of you smash the like button. If you want these notes, you will get all of these notes also. Okay. Yes, there are PYQs at the end of lecture also. I am giving you PYQs always. Okay. Now students, the next thing is called as selfing of F1. In selfing of F1, what do? You take the F1, cross it with each other. Now students, next thing is to make the gametes. How do you make the gametes here? How do you make the gametes? Always confusing, right? Capital R, capital Y, small y. How do you make the gametes here? Very simple, very simple. This with this, you need to multiply this with this and this with this, this with this and this with this. What do you end up getting, students? What do you end up getting? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. 4 different gametes, done. 4 different gametes, done. Now you need to do the cross. Similarly, you will have 4 gametes here. You will have 4 different gametes here. 4 different gametes, 4 different gametes. Then you need to do a cross. What is the cross? Capital R, capital Y, small r, capital Y. Capital R, small y first. Capital R, small y, small r, capital Y, small r, small y. From one parent, male parent. From female parent, what do we have? Again, same thing. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. You have to do entire thing. I will not fill it because it's very easy to do. How do you do this? Capital R, capital Y, capital Y, capital R, small y, small y. So similarly, you have to fill the entire table. Okay. When you fill the entire table, this is what we obtain. This you have to obtain. Now, once you obtain this, now you need to understand the phenotypic ratio and genotypic ratio. Okay. Now, students, what are total progenies here? What are the total progenies? Total progenies. Total progeny is 16 here, out of which we have round and yellow, round and yellow, round and green, wrinkled yellow, and wrinkled green. The ratio is very simple. The ratio is going to be 9:3:3:1. That is, these two are parental and these two are recombination. These two are recombination. Done. Okay. Now, what is the trick here? The trick is very simple. A dihybrid cross is always a cross between two monohybrid. Dihybrid is a cross between two monohybrids, right? So you can always do 3:1 into 3:1. 3 3s are 9. 3 3s is 9. 3 1s is 3. 1 3s are 3. And 1 into 1 is 1. Similarly, you can also find dihybrid. You can also find the genotypic ratio also. How do you find the genotypic ratio? This is the genotypic ratio in monohybrid It is the genotypic ratio in monohybrid cross. Just multiply it again. 1 into 1 is what? 1. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 into 2 is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 2 is 2. And 1 into 1 is 1. This is your genotypic ratio in the case of dihybrid. In the case of dihybrid cross. And with this, you get law of independent assortment. Now, what is law of independent assortment, students? What is law of independent assortment? Understand, very simple to understand, very simple to understand. Law of independent assortment. In F1 cross, in F1, did we have something like this? Capital R, small r, capital Y, small y? Yes. In this right here, do we have a pair of contrasting traits? A pair of contrasting traits? That is, two pairs of traits are combined here. One is for seed shape and other one is for seed color. Now, do we have here a pair of traits? What do you mean by pair of traits? Means totally 4 traits should be there. In seed shape, do we have your round and wrinkled? Here we have what? Yellow and green. Yes, yellow and green. So, law of independent assortment tells you that when two pairs of traits, two pairs of traits are combined in a hybrid, when they are combined in a hybrid, segregation of one pair, segregation of round and wrinkled and segregation of yellow and green is independent of other pair of characters. So, seed shape will separate out separately, seed color will also independently assort. That's all is law of independent assortment. The two pairs of traits will assort independently. That's all. See, independently they are assorting out. They are no blend, they are not mixing with each other. Okay. Understood, all of you? Understood? This is called as law of independent assortment. Okay. Seed shape and seed color will assort independently. Okay. All of you see this question. Okay. A yellow seed and violet flower pea plant are crossed with green seed and white flower. Green seed and white flower plant. The progeny showed 4 different phenotypes. This is the clue. 4 different phenotypes. The yellow seed and violet flowers are parent must be. What are the parents? Are they double homozygous, double heterozygous? What is the condition which you get here? Okay. Now I'll show you the first one. You see, if they're homozygous, what will happen? Okay. If they're homozygous, yellow seed and violet flower, right? Yellow seed and violet flower. If they're homozygous, if the parents are homozygous, and we are crossing it with green seed and white flower, right? Small y, small v. What are the gametes do we get here? Gametes. Capital Y, capital V. Small y, small v. And you cross it, you will only get one product. You do not get 4 different ones. Okay. You see this one. Double heterozygous. What is the double heterozygous? Capital Y, capital V, small v. And these are normal. Small y, small v. Now what are the gametes do we get here? Do we get 4 different one? Do we get capital Y, capital V? Do we get capital Y, capital V, capital Y, small v, small y, capital V, small y, small v? Here we only get one, that is V. small v, small y, small v. What are the offsprings? What are the offsprings? Capital Y, small y, capital V, small v, capital Y, small y, small v, small v, small y, capital V, small v, small y, small y. Once again, this is supposed to be y. Small y, small v, small v. Did we obtain four different phenotypes? Yes, we obtained four different phenotypes. So what should be the answer? Answer should be double heterozygous condition. Double heterozygous condition. Only in double heterozygous. Both are heterozygous here. Heterozygous condition. Similarly, students, try to make this one also. These two, you try. On your own, you try. This one also. See, homozygous seed color. Homozygous for seed color, that is capital Y. And heterozygous for flower color, that is capital V, small v. Try doing the gametes and crossing over. You will not get four products. You will not get four different phenotypes. Okay. Now, look at this question. What is the expected percentage of F2 progeny with yellow inflated pod in a dihybrid cross experiment involving pea plant with green color inflated pod, yellow colored considered pod? So what do we have, students, here? Parents is green color, green is dominant, pod color green is dominant, green inflated is dominant, recessive is what? Recessive is yellow color, recessive is yellow constricted. Students, is it not similar to your dihybrid cross again? Dihybrid cross again, yes. Similar to this one. Round in yellow, wrinkled in seed, wrinkled green. Similarly, we have something called as green inflated. So we'll also have what? Green constricted, and we will have yellow inflated. So how we have yellow, round in green, wrinkled yellow, wrinkled green, we have green inflated, green constricted, yellow inflated, yellow constricted. Ratio will remain the same, that is 9:3:3:1. Same ratio is maintained. Now they are asking you, what is the expected progeny of yellow inflated? That is yellow inflated, this one. So what is the formula? It's very simple. 3 divided by total progenies into 100. That is roughly around 18.75%, and this is the answer. So in examination they can always ask you, what about for green inflated, or they can ask you for yellow constricted, or they can change the entire characters. They can use flower color and stem height again. So all possibilities are possible, but the entire phenotypic ratio will remain the same only. Okay. Now there is a variation of such question, students. Sometimes what happens is they'll give you a question, but they will give you the progeny number. See, they're giving you progeny number. So in that case, what you should do? So they're asking you to find what would be the number of yellow wrinkled seed, right? Yellow wrinkled. So if you go to dihybrid cross, if you go, yellow wrinkled is how much? 3 again, 3. So what you need to do here is 3 divided by 16 into number of progenies, 800. That will be roughly around what? How much? 3 divided by 16. What is 3 divided by 16, students? What is 3 divided by 16, students? 3 divided by 16. I will use calculator, you can't use calculator. I am allowed. 3 divided by 16, the 0.187. 0.177 into 800 is around 150. This is 150. 150. So that's for today's class, students. That's for today's class. Thank you so much, and smash the like button. Keep coming live, and let us know in the comment section how was today's class. If you enjoyed today's class, let me know which part was very clear to you and you were able to solve some more questions. I believe you have an idea right now which type of question they can ask you also. One small idea is set in your mind right now. So with that being said, thank you so much, students, and I will see you in the next class. Till then, take care, and all of you. Bye-bye. Bye-bye. Bye-bye.